does a lot of machine learning, and just as open science happens, he was interested in dream challenges and just decided to participate. So he was a participant, but then he also helped a lot in the last part of the analysis. I have to say that I also have to acknowledge a couple of more people from IBM. Kelly Norrell, that helped with the scoring and was an essential part of the challenge, so please acknowledge them. And also Guillermo Cecchi, who's part of the neuroscience team in IBM, and a longtime friend and collaborator that also pushed us towards working on this olfaction problem that was very important. So Amit is going to give what we call post-challenge analysis of all the data that we had. I have to say that one more thing that we did for this challenge and that really showed that there's a very interested community in the olfaction was that we did a post-challenge community prediction that you will see the results of. So we had bi-weekly Google Hangouts where we discussed what else can we do in order to get better challenges and better results. And you'll see that the results were great. The community was very participative. So you'll see another results now. So Amit?
Thanks, Pablo, and thanks, Rick, for a great talk. So what I'll present now is essentially some of the insights that we gained from the challenge. As mentioned earlier by Leslie, that it was quite a surprise that many simple models, which also Rick mentioned, did quite well, and there were certain, I wouldn't say clear-cut, but quite strong messages in certain directions that we got from the challenge. So, like the 3 talks previously mentioned, so the challenge was essentially you were given around 500, a little fewer than 500 odd molecules, and you had 49 people who were random individuals, just commoners, and they were made to smell these 500 different molecules and rate them. Based on these 21 different dimensions, that is intensity, pleasantness being the most important, and 19 others which were essentially related to some kind of objects that we usually see in nature. So one of the key insights was that just based on structure, independent of genetic information or any other kind of information, the models were able to get a correlation which was greater than around 0.75, which is quite impressive. So the first thing, what we tried to see is, so in most cases, right, like in vision, so you have hue or you have RGB, we know that how much ever, say for example, red and blue you mix, you're not going to get green. So essentially that forms a basis for all kinds of colors that you can produce with these different 3 sets of colors. So the essence is that these 3 colors that we have are not correlated. So we tried to do a similar study in terms of the data set that we had, that given the 21 descriptors, were there any correlations? I mean, ideally you would want essentially a basis, mathematical basis, where you would have each of the features being independent and they cover the whole space. So as it turns out, it wasn't. So it turned out that, for example, sweet and pleasantness were very highly correlated. So, which is not very, I mean, which is kind of intuitive. Most people who smell something as sweet would say in very high cases that it was pleasant. Similarly with fruity and sweet. And people who said something that smelled acidy also smelled chemically, which is also not surprising. But it's interesting that this was all sort of data driven and it was all found through the data. And similar certain correlations. And it was found that, for example, the highest negative correlation was between musky and pleasantness. So people who said that something was highly musky didn't say that it was pretty low on the pleasantness scale. So we did, so rather than besides correlations, we were also trying to find if there were any causations that we could find. So we did a couple of things. So one of the things was sort of inverse covariance, but the other thing from data mining that you can use is, so we discretized the, essentially the predictors, the descriptors that were there into sort of binary categories. So say, for example, like above 50, if you predict, then it's a 1, otherwise it's a 0. And we tried a couple of these thresholds. And we ran these association rule mining techniques, which are essentially what they say is, I mean, they usually use it for market research. So say if you buy A and B, what are you most likely to buy? And that's how they stock stores and so on and so forth. So here we are trying to find similar things. That is, if people say, for example, something is fruity and sweet, does it relate to any other descriptor? And so we found that based on the previous things, that the highest sort of correlate, these causations that we found, the most sort of confident causations that we found, was that something that's fruity and sweet is most likely pleasant. And the interesting thing is, it's not the other way around. And this is also kind of intuitive to understand. So for example, you can have something that's flowery, which is pleasant, right? So you could, so you would say that it's pleasant, but it could be many more than just fruity and sweet. But if something is fruity and sweet, then with high confidence, you can say that it's pleasant. Also, the other interesting aspect here is that, so we tried similarly with intensity. And as also Rick mentioned in his talk, right? So you need almost all the 5,000 features to predict intensity accurately. And so it was the case here that we found certain rules, but it's extremely complicated. So you have like bunch of other descriptors coming in and bunch of other subsets that come in. And that's kind of, also you could explain intuitively, because intensity is not sort of specific to any kind of odor, right? You could create intense smells out of various combinations. I mean, I guess the parallel I could draw is with, say, sound, like amplitude. I mean, you can have loud music. It doesn't matter what the details of the song are. You can create loud music based on very different kinds of things. And so this is, and I think that's analogous to intensity. Also, the other thing is, I think among the 21 descriptors, probably pleasantness was the only sort of qualitative descriptor, in a certain sense, I would say. So for example, you had the other 19 descriptors, which are like woody, garlicky, which are related to objects that we find in everyday life, while pleasantness is something that's not specific to any of these objects. So it was kind of interesting that the highest sort of causation we found was with this sort of qualitative descriptor. Now, based on the inputs, as previously mentioned, so there were around 5,000 plus dragon descriptors. And what these are essentially, it describes the structure in terms of what atoms are there, how many atoms of a certain kind, and angles between bonds, and so on and so forth. So that's how the input data looked like. It was also interesting that if you just like ignored the outputs, that is the descriptors, and just looked at the input side, so you find very high sort of eigenvalue, eigenvector. The first eigenvector is sort of orders of magnitude higher than the other eigenvectors, and then it sort of decays. So I think many of the best performing models, it was more of a challenge about, unless your model is completely random, about capturing those lower eigenvectors in terms of capturing the signal. And it's also interesting, I mean, I think Pablo and others did a good job in dividing the training set from the final test set and so on and so forth, because it was kind of, from this you can see it was a representative. So this is with respect to the first sub-challenge where you had to predict individual subjects. And so what you see on the x-axis is essentially the team rankings. So there were around 18, 19 teams. And what you see on the y-axis is performance, so higher the better. And what you see on the, what you see the red line as is essentially the performance of the team. So obviously the first ranked team did the best and the last ranked team did the worst. And what is interesting is the blue line, which is essentially a wisdom of crowds kind of model. So what you're doing there is essentially, so for the first spot, you just take the best model. For the second spot, you take an average of the predictions of the first and the second model. For the third, you take an average of first, second, and third, and so on and so forth. And so what you see is that not only is the performance almost flat, you see that the best performance is around, say, 6, if I'm not mistaken. So around 6, you see that 6 teams that the performance, and it is better than the best performing team. So collecting different models and basically averaging their results leads to better sort of prediction. And this is not quite surprising from also a machine learning standpoint, because usually different models capture different parts of your space, which they call hypothesis space. And so aggregating these kind of gives you more sort of coverage in terms of specific predictions. This was a similar sort of chart, but with respect to sub-challenge 2. And again, here you can see the same kind of qualitative result. That is the wisdom of crowds model was much better. Now, these are some of the actual numbers from the first sub-challenge. But the thing that I want you to look at is that, so for example, like Rick mentioned, the first thing, the first winning model of UFAN was a decision tree, but the second one was a linear model, which was quite close in performance, which is very promising. And so some of the implications I'll discuss in the next few slides. But so this was seen for the first sub-challenge. And in the second sub-challenge, you can see the linear models are rank a little lower. But still, the scores are quite close. And the main result, actually, main reason, sorry, for this being true is because, as mentioned in the previous talk, predicting sigma is not linear. So if you use linear models for predicting that, that kind of tanked your overall accuracy by quite a bit. So what does this mean? So one of the higher level sort of insights, as I said from the previous two charts, is that linear models almost take you 95% of the way, which is very sort of promising in the sense that it leads to more sort of interpretable data mining. Also, a very interesting sort of application of this is that if you use a linear model and it takes you almost 95% of the way, you can invert it. So for example, not just taking you from structure to perceptors, but from taking you from perceptors to structure. So you could basically say that I want to create a smell which is, say, 50% pleasant, 75% intense, 30% woody. And you're given a bunch of molecules, you can actually solve an optimization problem and tell the person what's the best way of creating that particular smell. So that's a very sort of promising insight which can be used not just in one direction but also in the other. The other was that if you fit, even for the individual, for the first sub-challenge where you're trying to predict each individual, if you just fit independently different models to each individual, those results were not very good. The best models which won and did reasonably well were where you took a large fraction of the predictions from the mean model, and then you basically just sort of perturbed it with some sort of individual predictions from the individual models. So essentially, what this means is that though the challenge was to essentially predict, given a certain odor, generalize across other odors, this means that you can also generalize across people, because a large part of the predictions in many of these winning challenges was using the mean model or tying the parameter space together and getting the most accurate prediction. So even if you were not among these 49 individuals, what this means is that when you were, say today, you were told, okay, smell these same 500 molecules, it is very likely that we'll be able to predict your preference with high accuracy. So there is not only generalization across odors, but there's also generalization across people, which is one of the things I think that came out of the challenge, which is possible. Then, as mentioned in the previous talk, so pleasantness, which as I said is a very sort of qualitative descriptor of smell. The interesting thing was that though we had 5,000 descriptors, if you used too many of them, it actually reduced the performance. And with maybe like 10, 20 of them, you actually got much better performance. If you were highly selective in what you chose, it was quite good. So the thing is that, so it seems that pleasantness is sort of more simply modeled in our sort of biology. And so it seems to be more universal. Though the interesting thing is unpleasantness is not. So disgusting odors seems to be very cultural. So the negation of pleasantness is not unpleasantness, because there's a whole span in between which may not be in either direction. So I think one of the examples that I heard was some US trying to make these sting bombs, which they made and were like a complete failure in the Middle East or in some other areas. And the reason is because it's quite cultural. So then, as I already mentioned, intensity was quite high dimensional, and the reason is because there's so many ways of making something intense. I mean, it doesn't have to be any specific molecule. Then the other descriptors, you needed somewhere in between the number of features. So it wasn't as extreme as intensity, but it wasn't as low as pleasantness. The recent interesting insight, so I'll get to this in more detail, but was that we tried to see which were the molecules that were the hardest to predict. And we found that, and then we also tried to do some hypothesis tests in terms of trying to find out what features were different in these molecules compared to the other ones. And we found a lot of sort of these sulfur kind of features coming out. So we thought, initially we thought that maybe having sulfur is hard to predict, but that's kind of counterintuitive because if you have many molecules that have sulfur, they usually smell like rotten eggs or so on and so forth. So why is it that these molecules have sulfur and they're hard to predict? And it seems, actually, yesterday in an email, and thanks to Andreas and Leslie, so we found that the molecules actually that were hardest to predict had sulfur, but they did not smell like sulfur. So like they did not smell garlicky or they did not smell fishy. And that was one of the reasons why the models failed in terms of predicting them accurately. So this is kind of a interesting insight. So as I said, so then we tried to see which were the descriptors that were the easiest and hardest to predict. So it turned out that garlicky was the easiest, and I think that relates to sulfur and sulfur being present or not. And then the hardest was acid. That's yeah, I don't know why acid would be that hard, but it turned out because and in each of these cases, if you look at the variation, so it wasn't that acid had the highest variance in terms of the data that we had in terms of the descriptors. So all of these were somewhere in between. So there is something more going on there. So these among the odors, so the easiest to predict in terms of the mean were this, so which is like fruity grape, camphoras. The worst to predict, as I said, was L-cysteine, which has a lot of sulfur molecules, but it seems it has some rotten smell, but it's not garlicky. And the interesting thing is that in terms of standard deviation, the hardest to predict was water, which is odorless. So you'd think that people would just say it's odorless and just give zeros across the board, but I guess, I don't know if some people were just trying to find something in it and gave numbers which were quite random, because the standard deviation, if you take the descriptors, it's not close to zero. So that was kind of interesting. So then this is just another chart of the odors that were easiest to hardest to predict in the test set. So the x-axis is 69 odors and the y-axis is performance across the subject, and what you see is a 95% confidence interval. And so you can see the same thing for the individual models as well as the wisdom of crowds model. So the qualitative insight is the same, like you get a similar sort of qualitative split. And so this is just a different plot of the same model. And so you can see that there is an inflection point at the end. So like those 5 odors seem to be significantly worse predicted than the other 64. So if you take these and then if you do the same thing for the aggregated models, you can still see that you can find a different inflection point, but the key thing to notice is the last 5 in the individual models are the same last 5 in the aggregated models as well. So you do the worst on the same ones. Then what we did was by team rank, we tried to sort of plot the worst predicted odors and the others, and just trying to see if it's consistent across teams. That is the worst predicted odor for each of the teams the same, and if you draw a line, it essentially matches that. So the y-axis is the performance again. And if you do for the aggregated models, I mean, here it's like even a clearer split. So the worst predicted odors for all the models were essentially the hardest to predict were harder for all the models, even though they might be, in terms of numbers, doing a little better or worse. So these were the features that we found that were different from in the badly predicted odors and the remaining odors in the test set, as well as if you did sort of random sampling on the training set. And these were some of the features that came, which sort of repeated. And so you can see that a lot of them, if you see like a lot of them have this sulfur atom in them. And the reason turned out to be that for these molecules, essentially they just don't smell like the standard descriptors would. So there are a lot of very interesting open questions. What is the necessary and sufficient set of descriptors of human olfaction? I mean, that's a really hard problem. Maybe, I mean, listening to some of the talks, one of the ideas I had was probably maybe starting point is the 400 receptors, because if you have 2 completely different molecules, if they excite the same receptor to the same degree, then probably you smell it the same way. So that may be a starting point. But it's still interesting of how you would do it so that people can communicate it more easily, like RGB. Like I said, pleasantness seems more universal, but unpleasantness is not. So how do we model unpleasantness? That could be interesting. So if you have a bigger data set, maybe you can do more clustering and grouping and then build specific models. How much does adding other forms of information help? I'll just take you to some charts quickly because I'm almost running out of time. So And then, yeah, I mean, all these studies were based on pure molecules. So if you take mixtures, it'll be much more complicated because things can cancel out odors and so on. So this was from the collaborative phase. So in the challenge, only the dragon descriptors were given, which were basically, as I said, like angles between bonds. But in the collaborative phase, we used Morgan descriptors, NSPDK, kind of features, which are more sort of higher level features that give you the shape of the molecules. And they seem to improve the performance quite a bit compared to what was seen in terms of the best results of the challenge. And you can see that the top 4 is the highest result. And what it means, all but Gaber. Gaber is basically, I mean, he used a linear model. So adding a linear model actually helps to improve the overall performance. So it is capturing something that the other non-linear models aren't. Same thing for the second sub-challenge. And here you actually get a higher performance. And I think that's, again, related to standard deviation. So it's since it's highly non-linear. Yeah, so long-term, I mean, applications, there are a lot of applications that you can have, creating different smells and forming different kinds of optimization problems to figure out what's the best way to create a particular smell, forming bases of different smells, and so on and so forth. And that's it. Thank you.